Hi everyone, welcome to today's live webinar session on photoacoustic imaging of placental function. Today our presenter is Carolyn Bayer of Tulane University. My name is Christina Asa and I'm an application specialist here at Fujifilm Visual Sonics, and I'll be moderating today's session. At this point in time, I'd like to pass things over to our product manager, Drew Heinmiller, to introduce a topic that's upcoming for the company and very exciting and really relevant to today's presentation. Over to you, Drew.
Thanks a lot, Christina. I won't take up too much time, but I will note that Dr. Bayer is going to be presenting a lot of work that was acquired with our vivo laser photoacoustic and ultrasound imaging system. And the exciting news that Christina was alluding to is about our new vivo laser X system. So we're very excited. For those of you that know, our vivo laser platform, or for those of you who don't know, our vivo laser platform is based off our vivo 2100 system. We've now got a system, the release is imminent, the vivo laser X, which is based on the vivo 3100 platform. That way you get your co-registered multimodal imaging along with the vivo HD capabilities on the B mode. There's the touch screen, which really helps out with workflow. And we've got a new laser, as you can see, for those of you, again, who are familiar. It's much, much smaller. It's faster. It's more powerful, so we get more sensitive acquisition, better depth, better sensitivity, that kind of thing. And another additional wavelength range in addition to the one we have already. And another major change is separating out the light delivery from how we did it with the vivo laser, which is take the fibers and sort of put them in these jackets on the side so they can be removable. They can be interchanged to different sizes of fibers depending on the application. And finally, we've incorporated a lot of workflow enhancements, so really providing tools for molecular imaging, cancer, neuro, and cardiovascular applications that really help with workflow from image acquisition to quantification. If you're not familiar with photoacoustics, I believe Dr. Bayer is going to be talking about that today, so I'll hand it back to Christina to introduce our guest. Thanks for your attention.
Thanks so much, Drew. So as we mentioned, our speaker today is Dr. Carolyn Bayer. Carolyn Bayer is an assistant professor of biomedical engineering and Newcomb Fellow at Tulane University, whose research interests center on the development of novel medical imaging methods to study the dynamics of molecular expression and physiological function in vivo during pregnancy and development. Previously, Dr. Bayer was a National Cancer Institute F32 and Lymphatics Research Foundation funded postdoctoral researcher at the University of Texas at Austin, applying plasmonic nanomaterials as photoacoustic contrast agents to image molecularly distinctive signatures in breast cancer in real time and in vivo. She earned her PhD in biomedical engineering at the University of Texas at Austin, where she was an NSF IGERT Fellow in cellular and molecular imaging for diagnostic and therapeutics, and participated in the Nanoscience and Nanotechnology Portfolio Program. She has over 10 peer-reviewed publications, 2 patents, and has presented at numerous international conferences and institutions, including a 2015 World Molecular Imaging Congress Exhibitor Selected Poster Award from us here at Fujifilm Visual Sonics. So without further ado, I'm going to pass things over to Carolyn.
So thank you so much for the introduction and for the invitation to speak about my research in this format. So I'm going to be talking about our work looking at placental function and development and how we use photoacoustic imaging to accomplish those goals. Oops, that didn't work. Let's see how this works. Oops, why is that not working? All right, I'll do, that's very odd. Okay, here we go. Sorry about that. I don't know what is. Any hints here? Okay, well, hopefully I won't have to step through individually. It worked in our preview at the beginning. So I'm going to talk about why we image placental function, why we use photoacoustics to measure placental function, and then I'm going to talk about the specific results we have using photoacoustic imaging to image longitudinal development in vivo. And then I'm going to talk about two specific applications in placental function imaging, oxygenation, and transporters. So the placenta is a multifunctional, highly vascularized organ. It develops entirely de novo during pregnancy, so it's kind of unique in this way. It's used for gas exchange, nutrient and waste exchange, immune functions, as well as endocrine and regulatory functions. So it's also unique in that way in that it performs the functions of multiple organs. So during pregnancy, it undergoes extensive vascular growth and remodeling, and so that's one of the keys to understanding the function of the placenta. So there are some specific pathologies related to the placenta. I'm just highlighting a few here. First, preeclampsia. This is indicated by high blood pressure and protein in the urine, generally detected in the second or third trimester. Right now, they estimate this affects about 5% of pregnancies, but they do predict this number will increase. That means right now that 500,000 babies and 76,000 pregnant women worldwide die from preeclampsia or eclampsia every year. A second pathology of interest is gestational diabetes, which affects about 9% of pregnancies. Diabetes and gestational diabetes can cause insult and reactive oxidative stress generation in the placenta, leading to reduced growth and remodeling, leading to small for gestational age development. Lastly, congenital heart defects. Cardiologists have noticed that infants with congenital heart defects also tend to be small for gestational age. So again, that ties back to placental function and whether the placental developed, grew, and remodeled as was expected, and whether it was supplying the necessary nutrition for fetal growth. And congenital heart defects affect about 1% of pregnancies. So lastly, even if you're not interested in pregnancy and development, there's also the developmental programming hypothesis, where if you are exposed to unusual conditions in utero, that can impact your later health. For example, this was first noted when Barker observed that small for gestational fetuses also had an increased risk of cardiovascular disease later in life. So this is potentially a site of intergenerational transfer of disease risk, and so for many reasons it's of interest to study this organ. So the reason we're using photoacoustic imaging to study the placenta and the placental development, we can co-register the photoacoustic image with our ultrasound image. This is like ultrasound. Photoacoustics is non-invasive and non-ionizing. We can perform it longitudinally, which is particularly important for models like this where we are interested in tracking changes over time. We can image in real time. And additionally, I think important for this application is that I do see potential for future clinical translation. Photoacoustics, I think, would be great as a point-of-care bench-side implementation. The Maternal Fetal Medicine Clinic has expressed a clear preference for ultrasound over other imaging modalities. Photoacoustics fits in naturally with that same clinical paradigm. So in ultrasound, we use a high-frequency pressure wave, which is launched by the transducer and propagates through the tissue. As it travels through the tissue, it interacts with various interfaces in the tissue. It can be absorbed, scattered, or reflected. Ultrasound, which is reflected back to the transducer, is then received. And then it's processed using time of flight to construct an image of the tissue. Photoacoustics is similar, except it starts with a nanosecond laser pulse. So we use this nanosecond laser pulse to heat objects inside the body which are absorbing the light. When those objects heat, they expand, and when the very short 5 nanosecond laser pulse duration ends, the objects cool. And so that expansion followed by contraction sets off pressure waves which then propagate through the tissue, similar to ultrasound, and we can receive them with an ultrasound transducer and reconstruct the image similarly. So the key, what we like to say with photoacoustic imaging, it's key to remember that the transmitted energy in this case is light. So some of the inherent properties of light are that it is multiplexing and then it provides high contrast, like other optical imaging methods. However, the received signal is ultrasound. So we're able to achieve higher resolution at depth than optical techniques alone because ultrasound is not highly scattered like light. So our first foray into this area, we wanted to first establish what components of the developing anatomy produce photoacoustic signal. We knew some things about the size of the anatomy in our preclinical models, and we knew some things about when blood developed and when blood perfusion initiated, but we wanted to be able to demonstrate what we could actually visualize, what we could image with photoacoustic imaging. So that's showing, this is showing the results of that. So we had SWV mice pregnant, which were donated by my collaborator at UT, Dr. Rick Fennell. We imaged these animals from gestational day 8.5 to 16.5, and mice have a 19-day gestational period. So at 8.5 in our ultrasound image, we can see the uterine horn and decidua. We can also see the embryo, which is this hyperechoic circle that you can kind of see. 2 days later, at developmental day E10.5, we now can very clearly see an embryo, amniotic fluid, the uterine lining. And then 2 days later, we can also, at developmental day 12.5, we now begin to see the outline of the placenta very regularly and very clearly. We also see the fetus, the amniotic sac lining, and the amnion fluid itself. And again, continuing on, we also see outlines of the placenta as it develops and the fetus as it develops through gestation. Now looking at the second row, these are now photoacoustic image overlays over the ultrasound image, which is still in grayscale. And the red indicates the photoacoustic signal. So as expected, starting at very early stages in gestation, the uterine lining and decidua has an increased blood flow, and that's reflected as an increased photoacoustic signal in those regions. At the later stages of development, we see a strong signal in the placenta. Again, it's a highly vascularized organ, as you can see in the microscopy images on the right. It has a large amount of blood for that supply and for that function, so we would expect that we get a strong photoacoustic signal. So this was proof of principle demonstrating that we are able to achieve photoacoustic imaging of the conceptus and relevant anatomy over a longitudinal time span during development, and obviously all of these images were acquired in vivo and with the vivo laser. So that demonstrates photoacoustic imaging of anatomy, but what we really want to image is function. So here's how we accomplish that. Since we're using a tunable laser, we can vary the wavelength of the laser and acquire photoacoustic images at each of those wavelengths, and that's shown pictorially on the left. We compare, we use the equation that's shown to compare the photoacoustic signal as a function of wavelength to the fluence and the optical absorption of whatever absorbers we're expecting to find in that tissue. And we use that to find the concentration C. So the optical absorption spectra are wavelength dependent, and these can be found in literature. The photoacoustic signals are wavelength dependent, so by performing a linear regression or a linear fit of the photoacoustic signal to a combination, in this case, of oxy and deoxyhemoglobin, which we're expecting to be the major absorbers present in this tissue, we then can display a map of the oxygen saturation, which is that red and blue image in the center. So here are the longitudinal development results that we achieved using these methods. So we do see changes in oxyhemoglobin saturation over developmental stages, and we're looking at some various applications which I'll go into in a little bit more detail. So the first application I'm going to talk about deals with looking at hemoglobin oxygenation. I'm going to talk about placental ischemia and how it relates to preeclampsia, and all of that is using endogenous photoacoustic contrast agent. So placental function is an indicator of preeclampsia. So placental ischemia specifically is thought to be an initiating event in preeclampsia. So in preeclampsia, the clinical symptoms aren't detected until the late second to early third trimester, which is shown in this last line of this chart on the right, high blood pressure and protein in the urine. But what they believe that this originates with insufficient placental development in the first trimester. So the placenta is undergoing its major vascular growth during at the end of the first trimester. So the insufficient placental development that occurs at the end of the first trimester leads to reduced placental perfusion and placental ischemia. That placental ischemia sends out signaling molecules which induce endothelial dysfunction and lead to the high blood pressure and protein in the urine in the mother. So our question is, can we use photoacoustic imaging and specifically look at placental hypoxia and see if that provides an early indication of preeclamptic risk? So we're using photoacoustic imaging of oxygen saturation in a reduced uterine perfusion pressure model in the rat. So this is a Sprague Dolly rat, which is used for this particular model of preeclampsia. I like this image because we see this is a color Doppler acquisition with the vivo laser. So we see a lot of vasculature associated with the placenta and fetus. We can see 2 vessels of the umbilical cord. We can see some associated heart vasculature. And on the maternal side, we see the maternal uterine spiral arteries, which are feeding the placenta. So we use these animals, we're imaging these animals right now, and we're acquiring maps of hemoglobin oxygen saturation. This is an example shown on the left of our reconstruction. We are currently engaged in performing this reduced uterine perfusion pressure surgical model. It involves clamping the aorta and uterine artery, which feed the placenta. We clamp about 40% of the blood flow. That dramatic change in placental perfusion and ischemia induces preeclampsia in these animals, and it's been documented that 5 days later, if you measure their mean arterial pressure and protein in the urine, they display those characteristics of preeclampsia. So this is a well-established model of preeclampsia. And I think there is potential for clinical translation. The uterus at 12 weeks is about, the human uterus at 12 weeks is about 12.5 by 10 by 7.5 centimeters. State-of-the-art photoacoustic imaging depth, we say right now, is around 5 centimeters. So we're certainly in the same ballpark, and I think with some additional improvements to the photoacoustic imaging method, this could be something which could be applied as a screening method in the clinic. So next I'm going to talk about placental folate transport and specifically neural tube defect formation and how we can use exogenous photoacoustic contrast to study this particular problem. So folic acid supplementation is one of the major global health successes. About 20 years ago, global healthcare organizations began recommending supplementing our food and encouraging pregnant women to take folic acid supplements. And that has been very successful. It's reduced the incidence of neural tube defects by 40 to 80%. To give some numbers, that means that spina bifida, so spina bifida, one of the neural tube defects, went from around 2,800 babies annually to 1,500 babies annually, representing a healthcare savings of 400 to 600 million annually in the U.S., which is healthcare savings which would be required to care for these individuals with spina bifida. So obviously you can see that folic acid supplementation doesn't solve all of the problem. There is likely a strong genetic component. There are other environmental factors. So, and additionally, the mechanisms underlying how folate mitigates the development of neural tube defects are still under investigation. So, in my collaborators' lab, they study these mechanisms. They also use valproic acid. So valproic acid is a known teratogen. It's a pharmaceutical for epilepsy, which is known to increase the risk of neural tube defect formation. And they noted that placental cells in culture show a reduced expression of folate transport receptors after treatment with valproic acid. And so over on the right, if you can envision, there's a maternal blood pool, and it's being filtered and transported through various layers of epithelial cells into the fetal blood compartment. All folate is obtained through our diet. Therefore, any folate which the fetus needs, it's a critical molecule for metabolism and cell differentiation and growth. So therefore, any folate that the fetus receives must be transferred from the maternal blood into the fetal blood. So we want to answer the question of whether valproic acid also changes placental transport of folate in vivo, strengthening the concept that this could be a mechanism of valproate teratogenicity. So folate travels, as I mentioned, from the maternal blood supplied through the uterine artery through fetal-derived epithelial cells into the fetal blood supply. So these transport principles across these epithelial cell layers are similar to what you would find in intestinal or tumor cardiovascular models. And it's important to note that this effect is valid after the end of the first trimester when blood flow begins in the placenta, in the human placenta. And in rodents, it's around 10.5 days gestation. So our question is, can we develop a model to image folate transport? So I compare, so in this particular instance of photoacoustic imaging, we're using a contrast agent. So we're not just using the endogenous contrast that's present in our tissue. We're introducing a contrast agent. And I compare this to bright field microscopy versus fluorescence microscopy. Our standard photoacoustic single wavelength might be seen as bright field microscopy. You kind of see a single view of that system. But if we add contrast, we are targeting and we multiplex, we then can acquire a wealth of information about the composition of the tissue. So likewise, we can use contrast agents with photoacoustics for improved contrast, targeting, and multiplexing for molecular imaging. Right now, I am primarily using small molecules as the contrast agents. There is a large selection of near-infrared dyes. They've been developed for fluorescence microscopy. Therefore, they've been tested in many cases for toxicity both in vitro and in vivo. I found they often have improved biodistribution and perfusion in comparison to some of the nanoparticle formulations. Additionally, some of these molecules are available in the clinic, such as indocyanine green. So because of their chemistry, they can often be easily functionalized and targetable as well. The agent that I'm going to show in the work here was OTL-38. And this is a folate conjugated near-infrared fluorescent dye, which is being developed by on-target laboratories for imaging ovarian cancer, prostate cancer. Currently it's in, this might be slightly out of date, currently it's in phase 2 for ovarian cancer. The way this works, they inject this into the patient. The surgeon then performs surgery, uses a fluorescent light to locate the tumor, which is uptaken, preferentially uptaken, this folate conjugated near-infrared dye. So that's how it works for fluorescence, but for photoacoustics, we are using the non-radiative process rather than the fluorescent signal. So for photoacoustics, we want a molecule with a low quantum yield, but still high optical absorption. So as a first pass, we're looking at OTL-38 and seeing how well we can image this. So we combined our OTL-38 with folate receptor positive cancer cells. We incubated them, and then we rinsed them and incorporated the cells post-incubation in a phantom, a gelatin phantom. And then this is an image of the ultrasound in grayscale overlaid with the photoacoustic in red. And the photoacoustic signal is acquired at 775 nanometers, which is the peak absorption wavelength of the OTL-38. And so we can see that the folate conjugated near-infrared fluorescent dye does generate photoacoustic signal. So the next step was to see what we could see in vivo. And we injected this into a 14.5 days gestation SWV mouse through tail vein. Immediately after injection, we do see an increase in signal at the peak wavelength of the absorption of the OTL-38, and that's shown in the blue line. That's the skin. We don't immediately see a change in the fluorescence in the fetus, however, but at least we do definitely see immediately that we can detect the presence of the OTL-38 in the increasing photoacoustic signal after injection. So next we apply spectral fitting methods, as I've described, related to the oxygenation of hemoglobin. We do the same thing, only now we add in our other absorber, which in this case is our OTL-38, shown in green on this graph. We perform the linear fitting, since the photoacoustic signal is proportional to the optical absorption, therefore gives us the concentration. And so we get this estimate of our concentration of OTL-38 in the tissue. And so this is a video. It is going to be a little choppy because it's about a 1 second time lapse. It's about 1 second refresh rate on the frames, representing about a half hour on each image. And we do see at the very beginning that there isn't much signal in the placenta, and then as the frames progress, you begin to see some photoacoustic signal likely associated with some major blood pools or major vasculature in the placenta. And we also see an increase in the signal in the maternal skin, which is what we would expect. So we are able to distinguish in vivo the OTL-38 from the endogenous contrast. And just to verify that, we went ahead and did IVUS imaging with a caliper system, and we do see fluorescence in the ex vivo placenta and fetuses that have been injected, of the moms that have been injected with this OTL-38. And so this animal was sacrificed about 2.5 hours post-injection, and we do see significant fluorescence in the placenta and additionally some in the fetus. So this is our ex vivo verification of the maternal fetal transport of the OTL-38. So going forward on this project, we will be continuing to optimize our timing, our concentration, and to achieve increased photoacoustic signal intensity and increased sensitivity. We're also making some improvements to the contrast agent itself. And we plan to use this method to image changes in placental folate transport after treatment with valproic acid. The SWV mouse, when you inject it with valproic acid, it forms neural tube defects and cardiac defects. It is, however, rescuable with folate. So with an overdose of folate, it returns to normal phenotype. So this is a good model for testing our contrast agent and testing our photoacoustic imaging capabilities. So in summary, we're using photoacoustic imaging to provide high contrast, high resolution images of function. Specifically, we're imaging placental hypoxia to predict preeclampsia, and then we're imaging changes in placental folate transport. In this case, it's to look at teratogenetic effects of pharmaceuticals. I think this is something which has a lot of potential and a lot of possibility for expansion, and looking at the other functional functions of the placenta that I noted in the beginning. So I'd like to acknowledge funding from Tulane and funding during my postdoc. Most of the mouse data was acquired during my postdoc. And the Center for Emerging Imaging Technology S10, which funded the purchase of the vivo laser at UT Austin. And I'd also like to thank my laboratory and my collaborators, both those involved in the collection of this data and those involved in our future work with the reduced uterine perfusion pressure RAT. And I'd like to thank my graduate students and the undergraduates who also contributed to the work. I thank you for your attention, and I welcome any questions.
Thank you very much, Carolyn, for that excellent presentation. So at this point, I'd like to welcome anyone from the audience to please use your questions window in your GoToWebinar panel in order to pose any questions, and we'll translate those over to Carolyn. So we have had a couple questions come in so far. First, I'm just going to ask more on a technical note, is did you have any issues with the fetuses moving throughout your scanning period? I think you mentioned it was about 30 minutes for the scan.
Right. No, you definitely do. There's obviously fewer issues earlier on in gestation. If I remember correctly, in the mice, around 16.5 days, you do have significant fetal movement. We can reduce the number of wavelengths. There's a lot of people looking at that to optimize the fitting, introducing hardware which can acquire images faster in real time with multiple wavelengths also could go a long way to improving and reducing motion artifacts. There's in the models that we're looking at, there can be up to 14 conceptuses and fetuses. So sometimes if one is moving too much, we just look at another one, which obviously won't always be an option in clinical practice, but right now it's an option for us.
Carolyn, I'll just add to that actually the new laser, it's funny you mentioned imaging speed, but the new laser actually is capable of essentially switching wavelengths between pulses. So essentially you can do imaging at around 20 hertz. So you could do 5 wavelengths at 4 frames per second, for example, which could get around some of those motion artifacts where you're imaging with this much higher frame rate, so you can capture things between the movement as well.
Yeah.
Thank you, Drew and Carolyn. So another question we have here Can the fluorescently labeled folate rescue the valproate-treated fetuses?
Yeah, so that's an interesting question too. It's unlikely that we'll be using a concentration of contrast agent that is of the same order of magnitude as the folate itself, but it's definitely something that we'll have to account for in future experimental design and controls.
Actually, Carolyn, I have a question about just dosage in general. What sort of dosages do you need, for your, fluorescence imaging versus your photoacoustic imaging?
Yeah, so I haven't explored that to the extent that I'd like to yet in order to make conclusive statements about anything other than the dye, this particular dye that we've used in vivo. But I would say that we are likely to need an increased concentration. I started in the images I showed, I started with the concentration that they, for development they use IVIS. And so what they are looking at fluorescence for developing the OTL38. So I started with their initial starting concentration, and I would like a little bit more photoacoustic signal, so I probably will increase that once we have an opportunity to test another round of mice.
Thanks, Carolyn.
Mm-hmm.
So here's another question. How sensitive is this method in detecting tissue oxygenation? And the second part to that is, does the presence of major blood vessels affect the sensitivity?
Right. So sensitivity is really going to be very depth dependent for sure. So it's hard to assign a number to sensitivity. So would major vessels affect it? I think that certainly if you had a major absorbing vessel located immediately above what you were trying to image, it would definitely absorb some of the laser light. That's one of the advantages of the multimodal imaging. In practice, we'll often use the color Doppler to locate those major vessels so that we know what we're imaging. And hopefully in future work we'll be a little bit smarter about that, and we'll be able to accommodate and somehow use that in our image corrections, but right now we're not there. But certainly the presence of a major absorber above the images, the anatomy that we're trying to image, would influence the concentration, would influence the spectral properties that we're reconstructing, but using the color Doppler as we're positioning, I think helps us avoid those pitfalls as much as we can.
And another one here, how easy or difficult is it to analyze placental images using photoacoustics?
So, I mean, that's a pretty broad question. How easy? So, I mean, right now, this is an engineering system for us. We are taking these images and we're processing them offline. There are oxygen saturation measurements which are available in the vivo, and I simply haven't validated and compared our offline image processing methods to those available in the vivo. I don't know if, Drew, you want to elaborate on what's available in the vivo lab specifically, but there are groups, mine included, who are aware of the challenges in place making this a clinical real-time imaging system. And one of the collaborators on my list, Brian Suma, we're working to do some real-time image processing so that this is displayed in real time. So there are a lot of people working on that problem, and I think it's only a matter of time before the systems are easily used in the clinic and easily implemented in the clinic.
Thanks, Carolyn. I'll jump in as well, and I agree it is a somewhat of a broad question, but when you're talking about these photoacoustic images of oxygen saturation, First of all, some work has been done to validate these measurements. So for example, using an invasive method like there's this optical based system which detects like partial pressure of oxygen. So that's invasive, so you can put it into a specific part of the anatomy. And then we did this years back just for basic validation and compared it to our SO2 measurements, and they compared very, very well, nice correlation. But there was a study done a lot more recently out of Roswell Park Cancer Institute in Buffalo where they actually compared these measurements, these SO2 measurements, to blood oxygen level dependent MRI images, and again, got really nice correlation. So I know that doesn't necessarily speak to how easy it is, but I mean these images are being displayed, you know, and I'll say on the new system up to 10 hertz. You're getting this sort of real-time data, and then you have these images where you've got the oxygen saturation map displayed, co-registered or fused with the ultrasound image. So you know where the placenta is based on the ultrasound image. So it's easy enough just to draw a region of interest around that placenta, and then it just sort of automatically spits out an SO2 measurement. So I hope that kind of answers the question as well.
Mm-hmm.
Okay, we have a couple of questions here relating to other technologies. So maybe I'll group them together and I can split them apart later if you want me to. The first question was, have you done studies on assessing intraplacental oxygen heterogeneity and how it compares to other imaging modalities such as MRI? And then a subsequent question from another audience member was that they've been using nanoparticles and MRI to detect placental inflammation and are curious if the system can also be used to detect vectors labeled with nanoparticles. And they had heard something about using the use of gold.
Yeah, so the second question on nanoparticles, I mean, certainly I think the consideration would be ensuring that the nanoparticle isn't hindering the transport, right? So as long as you're able to demonstrate that the vector-targeted nanoparticle has the same transport, if that's what your question is, it's probably a little bit question-specific. But certainly, yeah, I mean, we extensively used gold nanoparticles looking at cancer, and they have a very strong photoacoustic signal, so they're also a good contrast agent for photoacoustic imaging. And then remind me the first question again?
So the first question was, have you done studies assessing intraplacental oxygen heterogeneity and how it compares to other imaging modalities such as MRI?
Right. So we haven't done that yet. I think there is one group that has looked at bold MRI versus placental oxygenation. But again, I don't think it was looking at heterogeneity. Obviously, we would expect heterogeneity. We would expect variation, overdevelopment, longitudinally. Also, there's likely some between animals. We worry somewhat about our anesthesia regimen, making sure that that's consistent so that the oxygenation of the animal itself is consistent. So there's a lot of potential sources certainly of heterogeneity, but that's also something that's interesting to explore and something that we can look at with photoacoustics that we often can't look at any other way. So
that's great, especially for such a lengthy question or a combination of questions there. So the next one is, what laser energy is used, and is there any consideration of exposure to radiation, as in the energy per area?
Right. So I've used a variety of systems, and I believe these systems were in the mid-teens millijoules per centimeter squared fluence, the laser that was used for the mouse images. Drew, maybe you can say what the current fluence is on your new system?
Yeah, the fluence is, I mean, it'll vary because we have different fibers for delivering the light, but it's actually, in general, it's actually higher than that, although there's easy ways of reducing it. But you've got the number right for the system that you used. I would say mid-teens, below the ANSI standard.
Right. So it's below the safety threshold for skin exposure. So we wouldn't anticipate any acute burns from radiation, from light.
So I think that somewhat answers another question here, but I'll pose it just in case you have anything else to add. Are there any known hyperthermic effects from using this technology, this approach?
Right. So, you know, there are some groups that have done, I know in my postdoc lab there was an effort to model the thermal characteristics of the photoacoustic signal generation itself. In general, it's perceived that the short duration of the laser pulse prevents bulk heating of the tissue. There may be some localized heating of the tissue. I did during my postdoc have a paper on heating cells with nanoparticles. So certainly with a high enough fluence, with the right exogenous contrast agent, you certainly could run the risk of causing either protein denaturization or some denaturation or some sort of effect on the cell. Generally, it's perceived that's going to be very localized, a very short, maybe on the order of nanometer influence or even smaller in temperature influence range, and bulk heating won't occur. But it's certainly, for contrast agents in particular, I think it's something that warrants further study still.
That's great, thanks. Someone else wants to know, have you also looked at fetal oxygenation? Is it possible to put these lasers on endovaginal probes to do human placental or fetal imaging?
That's what I'm hoping. Yeah, I mean that would be the clinical implementation. We would, for first trimester, the only way we would likely access the entirety of the uterus would likely be through some sort of transvaginal, or at least consistently be able to image the entire uterus. But certainly, yeah, there is a great interest in looking at fetal oxygenation, not just placental transport. Yeah, that's definitely something that I've discussed with clinical collaborators, and at some point in the future, hopefully we'll start looking at.
Great. Do you know if there's been any studies using photoacoustic imaging for visualizing placental margins? And maybe if not, could you comment on the use of photoacoustics for this application?
There is not. So this is, I mean, this is very new, a fairly new application. There's a large body of work using ultrasound for imaging small animals, and Doppler for imaging development in small animals and imaging placenta. But the photoacoustics, there's really just a few groups that have sort of begun dipping their toes into the water in this area. So there has not been anyone to look at placental margins at this point.
And for your work here, how frequent have you used this technique during the mouse pregnancy? So for your longitudinal studies that you showed, were those all performed in the same mouse?
They were. So I can't speak, I don't remember if those specific example images were all performed in the same mouse. But yes, we will image mice every other day, and we limit the duration of exposure to anesthesia to 2 to 3 hours per day. And we've had success with that, and we're able to image from 8.5 days gestation to 18.5 days gestation in the mice using that. There can be some fetal resorptions, so you definitely have to be careful, and it's probably species dependent and strain dependent to how sensitive they are It's not the imaging per se, it's the anesthesia duration and exposure that I think becomes of concern. So there is some risk there with the anesthetic regimen.
Yeah, maybe I'll just add to that a bit. Having done some pregnancy work in the past, I would agree that it is an issue of anesthetic and managing how long you want to maintain your dams under anesthesia. Generally, I've done many studies looking every 3 or 4 days of pregnancy throughout the entire pregnancy and not had any issues. So another question here, did you want to study and compare the reduced placental perfusion model with normal pregnant rats? And secondary to that, do you want to study another model, or are you interested in studying another model, for example, the DOCA model?
Yeah, no, for sure. So right now, Sun's photoacoustics and using photoacoustics of placental development is so new. I chose a model where we can clamp the blood flow and we know what we're changing. There are other chemical models, there are genetic knockout models. Certainly once we have a method that's established with our more mechanical model where we know we're getting the reduced perfusion, then yeah, we definitely would like to open it up to other models of preeclampsia. Mm-hmm.
That's great. We have an audience member here who's curious about any phenotypic abnormalities you saw in the animals. Did you see increased fetal death in I think it's the mice you did there, and they're mainly concerned about any changes or suspected changes, I guess, to sight or vision.
We didn't look at that. We didn't look at sight or vision because these were studies that were, they're terminal, right? We're not allowing the animals to deliver. We sacrifice at the end of our imaging session on the 18th day. So we don't have any way to look at vision. But again, I think that would be a factor of the anesthetic regimen, and depending on what you were trying to study, you would have to sort of do some trials and see whether your anesthesia did affect whatever variable you're trying to study.
Thanks. So this question was kind of brought up earlier, but I'll pose it anyways in case you wanted to add anything else. Has the technology been used in human placentas yet? And if not, what are some of the barriers you anticipate that could be preventing this in the future?
Yeah, no, this hasn't been applied to human placentas. I think the major barrier is imaging depth, right? So right now we can image, we say to 5 centimeters. In practice it's probably more like 2 to 3 centimeters depending on the system. So as we improve photoacoustic imaging technologies, and I'm saying that pretty broadly and intentionally because there are a lot of different ways we can do that, I do think that we'll be able to achieve more on the order of 5 to 10 centimeters of depth. And so no, it hasn't. So I think improving the technology so that we can achieve imaging depth, which is relevant to the human clinic, would be the first barrier. The second barrier is kind of esoteric, but just making it into a system which clinicians can be comfortable with. What's a little bit unique about ultrasound and hopefully photoacoustics is that it can be real-time at the bedside or bench side of the patient. And that is how the maternal fetal medicine specialists use it. I talked to one of them about this system and she immediately asked me, who's going to read the images? So she wants to be the person reading the images. She wants to have it there so that she can have the patient there. She can see the physical state of the patient while she's imaging and looking at either fetal oxygenation or placental function. So I think that making sure that it is not just an engineering tool, but something that clinicians can use on their own, I think is the second sort of major barrier.
We have one more, a little more technical question here. Do you think that the pharmacokinetics of folate are changed when you attach the dye?
It's certainly possible. That's why we're using a small molecule, so that hopefully those pharmacokinetics aren't changed. The OTL-38 has been tested in cells, and it has been shown that it does not change cellular uptake of folate that was tested in cancer cell lines. So certainly it hopefully will not change placental uptake. Certainly we'll test that in cell culture before making that statement. And then as far as changing other aspects of transport, it could, but that's why we're kind of hedging our bets and using a small molecule. And it's certainly something that we would have to study again.
That's great, Carolyn. Thank you so much. So that was our last question. I'd like to thank the audience members very much for attending our session today. And Carolyn, you did a great job with your presentation. I really enjoyed seeing your data, as I'm sure everyone else has. So just to...
Yeah, thanks for the opportunity.
Great. So just to wrap things up here, I want to invite everyone to stay connected with us here at Fujifilm Visual Phonics. Please visit our website. We have a lot of interesting information on there, even specific to both photoacoustics as well as developmental imaging. We encourage you to submit any questions you might have or support requests via web forms found on our website. We do have a full webinar schedule that will be upcoming, so please check back on our website in order to register for future webinars. You can find us on all social media channels. You can visit us at booths at conferences or congresses, and you can look on our website to find out which ones we're attending. And of course, if you have any questions about our products or technology, especially following up on this webinar, if you have any questions about our new laser technology that's coming out, we please encourage you to contact us using the customer support forms found on our website. So once again, thank you for your time and your attention, and we hope to see you again.